they affect different parts of the cell. And you'll learn this, okay? So keep your eyes open for that. This one here, this is really a straightforward, I want you to understand the relationship between the hypothalamus and the pituitary. Why? Because the pituitary is such an important endocrine gland. Okay, so it's just a matter of just go through the drudgery of learning about that connection and how the two are related. And then this one down here you'll see and hear today. Okay, you'll find out that there's something to do with this thing called the basilar or basilar membrane in the spiral organ. And it's something to do with how the basilar membrane is constructed. Is it really taut? It's a membrane now. It's connective tissue. And so it can be constructed in such a way that it's a very tight membrane or that it is a looser membrane. That very thing plays a role in the frequency of sound that we can hear and how we hear that frequency. It sounds totally off the wall at this point, but it will make a little bit more sense before we end today. Okay, when we get over in the lab, I will give you your lab test back. We'll start the lab with a dissection of the eye. We'll take a look at the sheep eye, deal with any other issues associated with the eye, and then we'll take a look at the ear and the tongue where taste buds can be located. Next week on Monday, it's all endocrine in the lab, and any kind of review that you want to do, I'll stay in the lab until 4:30 on Monday if you'd like to do some prep for Wednesday, and then that's it, okay? We decided last time, and I haven't heard from anyone telling me to change this, on Wednesday of next week, 10:15, we'll start our lecture exam. We'll go for an hour and 20 minutes. It'll then be over. I'll give you the rest of the time then between lecture and lab to prepare for lab, and then we'll take the lab exam at 12:30, okay? My other people who need makeups are not in the room. I just wanted to tell them that those makeups are going to start then after that lab test, so I guess I'll hold that for later. All right, so let's get back to then understanding a little bit about how the retina of the eye works. I would bet that if you were to get into a particular specialty where real intricate knowledge of the retina and how it functions is important, then you would go back and dig a little bit deeper. We're kind of scraping the surface. Where we are here is showing you let's say the chain of nervous tissue that are involved in the actual reception of light. Okay, we have learned that this thing is a rod cell. The generic term is a photoreceptor. It has the ability to detect the presence of light. And we found out that it is in these membranes inside this deepest portion of the rod cell that light can be detected. Light has to pass through, as we have learned, all of this tissue and eventually shine on here, and then certain molecules present in the membranes are going to respond to the presence of light. What you're seeing here, though, is something different, and this is why I present it. So far, we have heard that neurons, if they're not being stimulated in some way, are at rest. They're in resting membrane potential, and there is no actual potential being propagated. This one is an example of where a neuron or nervous type tissue is at rest, but it is depolarized at rest, which is different. It actually is in something called, it's generating what they call receptor potential. The rod cell is constantly in the dark releasing a neurotransmitter at its synapse with the bipolar cell. The bipolar cell is a type of neuron in the dark without the presence of stimulation. So it's a little different, and I won't beat this horse too much, but I just want you to see that distinction. Why? Because I want you to understand that there are exceptions, okay? And that it's hard necessarily to generalize everything. I mean, we can generalize, but then there's always exceptions in biology. Okay, back to here then. This neurotransmitter that is released, you will read about it, is a type of neurotransmitter that is an inhibitory neurotransmitter. So this bipolar cell is the type of cell that would normally just spontaneously depolarize. There are cells that do this. You will learn that the cardiac muscle tissue actually spontaneously depolarizes in the area of the nodal tissue. You've heard of SA node, AB nodes in the heart? Sarah has, right, Sarah? AB node, SA node, this nodal tissue, this muscle tissue literally spontaneously depolarizes, and it sets a pace. But anyway, this would be the type of cell that does that as well, unless it is inhibited. And the GABA neurotransmitter, the GABA neurotransmitter, inhibits this cell. In the dark, this cell does not stimulate that one. In the dark, this cell does not send an impulse to the neurons in the occipital lobe to tell them that there's light. Instead, the brain sees dark. In the light, that all changes. In the light, and we're going to see the mechanism, it's a G protein mechanism. In the light shines on here, and there's going to be some kind of a change that occurs in the membranes of this portion of the photoreceptor. And instead now, the photoreceptor cell is going to repolarize. It is not going to be generating receptor potential. It is not going to release an inhibitory neurotransmitter at its synapse with the bipolar cell. The bipolar cell will then spontaneously depolarize, okay? Expect it to be fairly leaky to sodium ion, okay? And then it is going to release a neurotransmitter at its synapse with the ganglion cell. The ganglion cell will send action potential to the primary visual cortex of the occipital lobe. Okay? This is well, we're going to see the mechanism here in just a minute. This is the photopigment. Okay, you've learned about this already. You've learned that the protein portion, opsin, combines with retinol to form a purple-looking stuff called rhodopsin. By the way, when we cut open the sheep eye, you're going to see like a purple liquid in the pan as you start to see this photopigment. Okay? All right, so when the light shines on the rod discs, then there is a change in this protein, as you know. The retinol changes shape, and it can no longer remain compatible with the opsin, and it separates. Okay? We see that here in this cyclical diagram. It separates. Okay? If the light stays on and stays bright, then we'll get complete bleaching out of the photopigment, and the rod will become inactive. Okay? But if the light, shines and then, it rises and falls, intensity rises and falls, then you'll see this thing cycle back and forth, and the rod is always ready then to detect light. What is the mechanism? I know you're going to love this, okay? You've seen this sort of thing before, but now we need to get into the nitty-gritty of it, and when we look at the endocrine system, we'll revisit it again. So, as biologists, junior biologists, whatever you want to call yourselves, you are seeing that this is going to be a very important mechanism in many cells. This is in the rod cell. There's our rhodopsin, okay? And we are seeing that there are proteins either in the membrane or near the membrane, just under the membrane surface, okay? So these things are floating around in here, all right? And they're bumping into substances on the inside of the membrane. This substance here is also present in the cell, bouncing around. You know that, perhaps. You know that molecules always are in motion. So they're always bumping into one another. This substance here is called cyclic GMP. GMP is a lot like ATP. The P stands for phosphate, just like the P in ATP. The M, though, stands for mono. So this is a monophosphate, not a triphosphate. The G stands for one of the nitrogenous bases that are included in DNA. Adenosine is for adenine, which is one of the bases in DNA. Guanosine is for guanine, which is one of the bases in DNA. Why do I tell you that? Because these molecules are somewhat related to the nucleic acids. Okay, so this thing is what's going to be called an internal messenger, okay? And it's always present, but its concentration will vary. And the more of it is, the more of it that's present, the more the message, the less of it that's present, the less the message. Now let's see if we can't link these together. This is in the dark. Let's ignore this photon of light for right now. This is the way it looks in the dark. There's our bent retinol. Now it's in the light, and the retinol has changed to transretinol, and something has happened. This opsin protein has changed its shape, okay? And it has become an enzyme, which is going to lead to a conformational shift on this thing here called transducin. Transducin is called a G protein. It's an internal messenger. When we mess around with transducin and change its shape, it turns it from off to on. And look what transducin does then. Transducin serves as a messenger to activate this enzyme. Am I that concerned that you memorize the name of this enzyme? No. On the other hand, you will encounter it because it is a common enzyme in cells, and the G protein is a common mechanism. Anyway, to get back to it, it's called phosphodiesterase. Okay? So what this enzyme does is it changes the form of GMP from a cyclic GMP to a non-cyclic GMP. That is not earth-shattering, but in the chemical world it makes a difference. This enzyme is present in the membrane of the rod cell, but it is inactive until this molecule is changed by the presence of light. When this molecule is changed by the presence of light, the transducin is changed, and it can now interact with inactive phosphodiesterase to make it active phosphodiesterase. Hang in there, okay? We see over here there are some ion gates, okay? So this is highly dramatized, okay, membrane. Here the gate is closed, and we see that there is a binding location for cyclic AMP. The gate is closed. There are no ions coming into the rod cell. Here, the gate is open because we see that there is an association between cyclic GMP and this ion gate. So obviously we have a ligand-gated ion channel here, okay? And the ligand is cyclic GMP. The ion gate is open, and we're seeing not only sodium coming in, but we're seeing calcium coming in as well to depolarize the cell. Okay, when phosphodiesterase becomes active, it begins to catalyze the degradation or change of cyclic GMP to GMP. Why is this important? Because only cyclic GMP fits into the receptor location on the ion gate. As this substance declines in concentration inside of the cell, it cannot actively fit into receptor locations, and the gate will eventually close. So what we have learned is that the rod is constantly depolarized in the dark. And here is the reason. Because there's plenty of cyclic GMP under the membrane to interact with the ligand-gated ion channel. But if cyclic GMP's levels come down because this has been activated by a light reaction, then cyclic GMP cannot fill this space, sodium and calcium ions cannot come into the cell and depolarize, and the cell repolarizes. Okay? The interesting thing is, well, how the heck did this ever come about? Okay, and I can't tell you that. Because it is a different way to get at the same thing. Okay? And the same thing is to not have action potentials go to the brain when there's no light, right? If there's no light, there's nothing to transmit. So we are still using a system where we're going to activate a particular neuron when light is present, but we're going to do it by deactivating another cell. It's sort of weird. Do you want to talk about this anymore?
Is this one of our essay questions?
No.
Okay.
Doggone it, I should have made it.
Then no, I don't want to anymore. I thought it was for a second.
This up here is something that the author of the book put together, just help you understand the way things work, okay? So we had our receptor, which is, in this case, the receptor for light now. It's not ligand gated, it's